So thank you for the invitation. I am Benoit Hedan. I am a vet in the canine genetics team, led by Catherine Andre in Rennes. And I will focus my presentation on the interest of somatic mutations and liquid biopsy to improve the care of histiocytic sarcoma. So you probably know that histiocytic sarcoma is a tumor coming from histiocytes. So histiocytic cells could be monocyte, macrophage, or dendritic cells. And the tumor can be in human or dogs localized or disseminated to different organs. So skin, joints, spleen, liver, or lungs. And it's a very aggressive cancer with a very limited response to chemotherapy. In humans, hopefully, it's a very, very rare cancer. We estimate that in France there is between 1 to 5 cases per year, so it's very rare. Half of the cases are secondary to lymphoma or leukemia. This is different from what we observe in dogs. And it was shown that there is a transdifferentiation between lymphocytes to histiocytes in this cancer in half of the cases in human. But the remaining half of the cases in human are close to what we observe in dogs. And due to the rarity of this cancer, there is no consensus on the prognostic factors and on the standard treatments. So there is a huge need of model to improve the cares in human medicine. In dogs, this cancer is also very rare in the whole population, but very frequent in few predisposed breeds. Bernese Mountain Dog, Rottweilers, and flat-coated Retrievers are highly predisposed. And in these predisposed breeds, we observe a familial transmission of this cancer. So we can collect numerous cases in the same family. Here you have a pedigree of Bernese Mountain Dog with affected dogs in black. And you have also dogs affected by lymphoma in blue and mast cell tumor in greens. And you can see that we can follow the cancer, the transmission of cancers across generation. So we hypothesize that dogs model could be a unique model to identify genetic basis of this cancer in human for human medicine. And indeed, the clinical presentation observed in dogs recapitulates what we observe in human. The tumor can be localized to one organ, most frequently the skin of the joint, and this is frequently observed in retrievers. Or the tumor can be disseminated to different organs, and we observe that more frequently in Bernese Mountain Dog. And recently, neurological form of histiocytic sarcoma has been described in Corgi. We have a different hypothesis to explain this different clinical presentation. First, it could be the same cancers observed at different time. So the localized histiocytic sarcoma to external organs are diagnosed earlier than disseminated histiocytic sarcoma to internal masses, which are diagnosed at the time of dissemination. Or the second hypothesis could be that the different tumors involve different subtype of histiocytic cells. So it could be different subtype of histiocytic sarcoma. So the long-term goal of our team is to identify genetic mechanisms involved in histiocytic sarcoma. We focused on the identification of germinal mutations, so the predisposing mutation, and for that we are working on with blood samples of affected and unaffected dogs to identify the predisposing genes. And this work has allowed us to develop a genetics tool for breeding selection for Bernese Mountain Dog, and I will not focus on this aspect. And the second aspect is a focus on somatic alterations. So the germline mutations, predisposing mutations, are not enough to develop a cancer. And during the life of the dog, there are accumulations of somatic alteration in specific cells leading to the cancer. And the second axis of our project is to identify these recurrent somatic alterations linked to the tumor progressions. And you will see that it's interesting for the diagnosis and the treatment of this cancer. So we focus on, for the somatic alterations, we focus on the MAP kinase pathway in histiocytic sarcoma. Indeed, we and other people have shown that over half of the cases of histiocytic sarcoma are mutated, activated in the pathway, the MAP kinase pathway. This pathway is activated in numerous cancers in human and dogs. And with these activations, we have the increase of cell proliferation, cell survival, and invasion and metastasis. So in dogs, in canine histiocytic sarcoma, this pathway is activated in over 64% of cases. And we have shown that there are recurrent mutations in an oncogene called PTPN11 in over half of the cases. So you can see here that over half of the cases are mutated in these genes, but we have other mutation in the gene named KRAS or BRAF, so exclusive mutations with PTPN11. So all mutation in these oncogenes lead to the activation of this pathway and explain a part of the development of histiocytic sarcoma in dogs. And interestingly, when we look where the mutations are found in the canine proteins, you can see the distribution here in red in the canine histiocytic sarcoma. And if we look at human histiocytic sarcoma or human cancer, in blue, you can see that we observe exactly the same hotspots, the same two main hotspots in dog and human cancer, leading to the activation of this pathway. So here we can see that the role of the protein PTPN11 is conserved between human and dogs, and activating mutation in these two hotspots are leading to the same effect, are leading to cancer. We have also shown that these kind of mutations of PTPN11 are enriched in histiocytic sarcoma, disseminated histiocytic sarcoma, so in dogs and also in humans. And we and other people have shown that these mutations of PTPN11 are not found in other frequent cancer of Bernese mountain dog, not found in lymphoma, melanoma, or mast cell tumor. So we hypothesize that this kind of mutation, PTPN11, could be very interesting to improve the diagnosis of histiocytic sarcoma. And for that, we focus on the liquid biopsy. So as you probably know, it's when a cell is dying in the body, the cell releases DNA in the blood. And if the DNA comes from a tumor cell, it's possible to detect the tumor DNA by looking at specific mutations, specific tumoral mutations. And we use this, the interest of the liquid biopsy to explore the interest of this method to do the diagnosis of histiocytic sarcoma with PTPN11 mutations. In human, the liquid biopsy could be very interesting for different applications, for the diagnosis with liquid biopsy, or for also an early diagnosis. They are useful for the prognosis, but they are also interesting for to monitor the response to treatment or to select new treatments. And I will explore during the next slide the interest of somatic mutation of PTPN11 in histiocytic sarcoma, and how we could use it, this kind of mutation, to explore these different aspects, different interests of liquid biopsy. And to detect PTPN11 mutations in plasma, there are different techniques. We could do next generation sequencing. Here we focus on a technique called droplet digital PCR. So it's a very sensitive technique to detect rare mutations. And to do that, we have a specific step where we have emulsions making 1,000 droplets, and each droplet is one reaction. And with this kind of partitioning with thousands of droplets, it's possible to detect very rare event, and it's possible to detect rare mutation of PTPN11 in our samples of plasma. So the first step of our work was to look if we could use the somatic mutation of PTPN11 to make the diagnosis of histiocytic sarcoma. So to do that, we selected 45 dogs with histiocytic sarcoma. We sequenced the tumors, and we found that over half of the cases half of the tumors were mutated for PTPN11. And then we look for the presence of the mutation in the plasma. And you can see that we detected in 21 of the 23 cases mutated, and we never detected in the cases with wild-type tumors. In parallel, we checked in a control population of dogs with lymphoma, melanoma, mast cell tumor, or healthy, if we detect this kind of mutations in the plasma. And we detect only one false positive, which was old Bernese mountain dogs, 10 years old, euthanized for another reason. And we can't exclude that the dog was developing cancer, but we conclude that we have one false positive with the method. And so using PTPN11 mutations to diagnose histiocytic sarcoma on liquid biopsy, we can conclude that we have a sensitivity of 33%, which is logical since the mutation is present in only half of the cases. And this sensitivity increased up to 77% when we have a lung localization. And this is explained by the fact that this mutation is linked to disseminated histiocytic sarcoma. And so when we have a lung localization, we are more likely to have a mutated histiocytic sarcoma for PTPN11, and we are more likely to detect it in the plasma. And we have a very good specificity of 99%, meaning that when we detect it, we are almost sure that we are facing histiocytic sarcoma. So this was published recently, and we think that this tool could be very interesting for the diagnosis of histiocytic sarcoma, especially when we have non-biopsiable masses. So now, since we have proved that it's possible to use PTPN11 to do the diagnosis of histiocytic sarcoma, we were asking if this marker could be interesting for the prognosis or for an early diagnosis. So for the prognosis, we know that we have a different prognosis for histiocytic sarcoma if it's localized or disseminated. The disseminated histiocytic sarcoma are very more aggressive with a shorter median overall survival of, in average, 2 months, while localized histiocytic sarcoma have a significantly longer median overall survival of almost 1 year. So, and we know that the mutation, PTPN11 mutation, are enriched in histiocytic sarcoma, disseminated form of histiocytic sarcoma in canine and human. So this mutation should be a prognostic interest in canine histiocytic sarcoma. And in human, we know that this kind of mutation could be a prognostic marker in pan cancer. So we are doing right now a retrospective study on histological block of histiocytic sarcoma from different vet lab in France. It's just ongoing, so we have results on the frequency of the mutation, but we don't have results on the link with other prognostic factors. And in parallel, we are doing a prognostic ongoing prospective study with oncologist vet from different localization in France. And we are collecting blood samples of dog at the beginning of chemotherapy to see if the detection of the plasma of this mutation in the plasma is linked to the overall survival of the dog or not. So it's preliminary data results on only 9 dogs coming from these different vets, and I thank them for this help. But you can clearly see the difference of overall survival of dogs with the presence of the mutation in the plasma at the beginning of chemotherapy in comparison of dogs where we don't detect the mutation in the plasma, which have a longer overall survival. So we have to do this on more dogs to confirm the preliminary results, but I'm confident that we will have a link between the overall survival and the detection of this marker at the beginning of the chemotherapy. The second aspect that we want to explore is the interest of this liquid biopsy diagnosis for an early diagnosis. So are we able to diagnose histiocytic sarcoma before the onset of the cancer? And to do that, we started a 2 years study with a vet student in the vet school of Nantes. And we follow up 30 dogs from 2 breeders. So with the 2 students, we take blood samples every 3 months. We follow the dog until death. And if the dog died from histiocytic sarcoma with mutation of PTPN11, we were able to come back to the plasma samples and look at the presence of the mutation in the plasma. So after 2 years, we observed 9 deaths. 6 deaths were due to histiocytic sarcoma with PTPN11 mutations. And you have here the results of the detection of the plasma before the onset of the cancer. So for the first dog, we were able to detect the mutation in the plasma only 1 month earlier the onset of the disease. But for some other dogs, we were able to detect the mutation 11 or 14 months before the onset of the cancer. And if we focus on this dog, we performed 2 months earlier the onset of the cancer. We performed X-ray, CT scans, and we detect nothing, while in the same times, we have been able to detect the presence of the mutation in the plasma for 9 months earlier, for 9 months. So we can conclude we can hypothesize that the liquid biopsy will be more sensitive than X-ray or CT scan to detect the cancer. And in average, with this preliminary study, we are able to diagnose histiocytic sarcoma in average 6 months before the onset of the cancer. And this number of 6 months is very interesting because in another study from in an American study where they focus on different cancer on a large cohort of 1,000 of dogs, they focus on different cancer by next generation sequencing. So they don't use ddPCR like us, and they don't focus on specific mutation, but they look at all many different genes. They were able to diagnose an hemangiosarcoma 6 months before the onset of the cancer. So I think that for hematopoietic cancer, such as lymphoma, hemangiosarcoma, or histiocytic sarcoma, we could use this liquid biopsy for an earlier diagnosis, and we could expect that we could detect this hematopoietic cancer in average 6 months earlier than the onset of the cancer. And finally, this liquid biopsy and the somatic mutation could be very interesting for the treatment selection and the treatment follow-up. Knowing that histiocytic sarcoma proliferate with activation of the MAP kinase pathway, give us opportunities to use a specific treatment, so targeted therapies. And we know that in human medicine, there are specific inhibitors of the MAP kinase pathway. In the lab, in parallel in the lab, we have developed 13 cells of histiocytic sarcoma. All the cells are mutated for PTBN11, and we tested this kind of drugs, so targeted therapies targeting the MAP kinase pathway, and we have shown that all the cell lines are sensitive to these drugs in vitro. And in parallel, the work of Takada et al. Have shown that the canine histiocytic sarcoma in mouse model are sensitive to this kind of inhibitors. And now there is a pilot study, a trial in dogs to test this kind of inhibitors, MEK inhibitors, so drugs targeting the MAP kinase pathway to treat canine histiocytic sarcoma. So if I summarize the interest of somatic mutations, they have many interests in human medicine, going from the diagnosis to the treatment follow-up. And using liquid biopsy, we have shown that for histiocytic sarcoma, it's possible to use that for the diagnosis, also for the prognosis, and also for the early diagnosis. I hope that results on the pilot study will show that this marker will be interesting for the selection of treatment and as in human medicine. And I have to thank for this work many people, especially the team, the canine genetic team, different collaborators from different vet schools, and especially Jérôme Abadi from the Nantes Vet School, who is working with us on this project for over 20 years, and the French vet oncologist who collaborate with us to look on the interest of liquid biopsy for the prognosis. And I have also to thank people funding the foundation to fund which have found this work, and all the owners, vet, and breeders who send us samples. So thank you for your attention, and I will be happy to answer questions now.
Thank you, Benoit. That was truly inspiring. I'm going to now stop recording because I noticed, or someone told me that people cannot use the Q&A. So I'm going to let everyone in as contributors, but I don't want people to then have their faces on the recording. So I'll stop now.